Hello everyone, how are you all? कैसे हैं आप सभी लोग? I hope you all must be having a great time. This is your faculty Ankit Rothan, and now we are going to start paper number 2, that is unit number 1. And over here, this is all about cell biology and genetics, the most fundamental unit for your paper number 2, guys, for your agri optional. And not only for that, but also its utility you will be seeing in a prelims and GS3 mains also, because over there one portion is science and tech which you have to prepare. Under this, the major portion which comes is from biotech. So this will be sorted if you will be understanding this as it is, the way I'll be telling you, with the linkages, not only with paper 2 but with paper 1 of agri optional also, with prelims and GS3 mains also. I'll be covering your biotech for all these particular aspects. Right now, before proceeding ahead, I have a lot of students who are not having agri as their background in their graduation, also who were not having this science background. So you don't have to have this barrier that you are understanding anything technical or science based things right now. Right, the way I'll be telling you, you have to absorb this, because ultimately your goal is to become a civil servant, not to become a botanist or to agriculturist or a genetics person, right? So we will be understanding it in a very scientific way with respect to UPSC, that how it is coming in a UPSC, that I'll be telling you. And if you are connected already, you must be knowing, but I'm just boosting you so that you are starting a new paper, and this paper carries a huge weightage now, altogether 300 marks, right? So fundamental starts from here. Slowly and slowly, sit back, keep your notes with you, keep your pages or register which you have made with you. I will suggest you to make A4 sheet size paper over here, right? Keep this with you so that it will be easy for you to handle all the notes which you will be creating. Okay, so with this note, let us start. And as we start with the syllabus which is written in our UPSC notification, you will be finding this paragraph is for cell biology and genetics. As you know that this heading is not written, but when you will be understanding all these topics, this is basically academically we are learning the cell biology and genetics only, the most fundamental unit, again I am saying. So I have divided all these lines into certain topics, and you will be finding there are all total 9 topics which are relevant for the examination from here. And let me tell you, this is the most asked unit also. At least 3 questions you will be finding in the paper number 2, at least 3 questions you will be finding. And as I have told you earlier also, all the FC students, I have told you that in paper number 1 you have to fix your 6 units out of 8 units are there in paper 1. You have to by heart do 6 units. In paper 2 now it's come you will be seeing that total 7 units are there. Out of this you have to do 5 units by heart, and out of this 5 unit, this first unit which we have started is one of the most important unit which you will be doing by heart. That means every line of the syllabus, every PYQ which is being asked, we will be handling that, right? Also you will be what you will be doing, you will be clubbing this particular lecture series with the enrichment lecture too. Parallelly, you have to go through the enrichment lecture. And the importance of enrichment lecture, I have told you, you have to attend this live if possible, if that is matching with your schedule, right? Because in enrichment, I let you do this active recalling. We utilize this active recalling method so that you can remember things in the class itself. So I'll be helping you out with this particular session only. But enrichment is furthermore, now we go one step ahead, where I'll be putting extra effort and you will be putting extra effort. Understanding, guys? So now let us begin. This is the build up for this particular topic which we are going to start. One by one we are going to pick the topics, and our first topic is going to be cell structure, function and cell cycle. Very important. One question is fixed from this particular topic. Because I have told you, out of all these units or out of all these subunits, certain subunits carry huge weightage, and this is one particular topic which carries huge weightage. Every year you will be seeing one question fixed from this particular topic. From a rest you will be seeing alternatively. Like this year they have not asked mutation, heritability, cytoplasmic inheritance. They have asked from here. Alright. So like this you will be finding that this is a fixed unit, fixed lecture, from where one question is definitely going to come, whether in civil services, Indian forest services, or in your state services. ठीक है? So cell structure and function, मतलब क्या समझ रहे हो? What you are understanding by it? That means over here you have to understand about the cell and a different organelles, their structure and function you have to focus. Your note should be highlighting these two things. Okay? And when we will be analyzing the PYQ, you will be finding this only. Let me first show you the PYQ so that you can understand. Dekho, these are the 2025 recent. This year only they have come, 2025. Define cell cycle. Directly what they have asked over here, cell cycle. Then function of plant cell wall. Structure of chloroplast. Structure of plasma membrane structure over here. Function of Golgi apparatus, endoplasmic reticulum. What you are understanding that whatever cell organelles we are having, their structure and function are being asked frequently. Every year they will be asking. And this cell cycle is coming 2 or 3 year gap. You will be finding that this cell cycle question is coming. Alright. So what will be our objective of this particular lecture? Objective will be, first you will be understanding over here about the cell theory. In the cell theory, I will be telling about 2 things, guys. One, a classical theory. And second, over here, we will be learning about the modern theory. Classical theory means when these scientists first thought about that, what is this? Then what they were thinking about that particular aspect and what laws they give with respect to that particular classical theory. And when modern instruments came into picture like electron microscopy and other modern equipments you will be finding, then we explored more and we got to know about the cell system actually. We understood that there are 2 different types of cells over here. So types of cells you will be understanding over here. And in this you will be understanding that what is the difference between a prokaryote and a eukaryote. I will be also highlighting the difference between a plant cell and an animal cell. And our focus will be entirely on the plant cell. Right, then we will be opening up this plant cell over here. This is the most important thing from where question will be coming. Plant cell, and here question will be coming, most important. And in plant cell, we will be understanding different organelles which are present, guys. So over here, our objective is to understand the organelles. Single M. Okay, organelles you will be understanding over here. What in organelles you will be understanding? 2 things, their structure and function. यहाँ से question आने वाला. From here you will be going to see the question, structure and function. Let me tell you, this lecture will be enough for you to handle any type of question which is going to come in the examination. Okay, that I am telling you that people have utilized also and scored well in the exam, mains examination. You will be scoring well. Plant cell is going to be the most important. This will be our objective, and then we will be understanding, not in this particular class, but this will be connected over here, cell cycle. To understand cell cycle, there are a lot of things which you need right now. So cell cycle we will be understanding at the right point of time. This whole plant cell, understanding plant cell will take us a lot of time, right? So our objective is huge in this particular class. That is, we will be learning cell theory, types of cell, plant cell. I hope you are connected and you should be very focused over here. I already speak or teach in a good pace over here, so no need to do it in 2x, rather do it in 1.5. It will be good for you. 2x पे पढ़ोगे तो कुछ समझ में नहीं आएगा, right? If you are watching it in a recording form. ठीक है? So these are the objectives over here. So what under organelles you have to understand. इसको भी आप explore करो कि organelles में हमने क्या क्या समझना है, ठीक है? ये हमारा सबसे important part रहेगा. तो organelles में आपको समझने वाले हो, यहाँ पर आप समझोगे just a little bit. You will be understanding about the cell wall. You will understanding about the plasma membrane. You will be understanding about the mitochondria. You will be understanding about the plastid and underplastid chloroplast. You will be understanding about the endoplasmic reticulum, Golgi apparatus. You will be understanding about the ribosome. That means over here 1, 2, 3, 4, 5, 6, 7. 7 organelles you have to focus upon, and there how you will be focusing upon structure क्या है sir and function क्या है. What is the structure? What is the functions? Structure naming you should know so that you can draw a schematic diagram. A diagram should be there. Functions, 5 functions you should know, not more than that. Either the question will be two pager means 10 marker, or it will be a 3 pager. 3 pager means 15 मार्कर और 20 मार्कर ओवर है। 20 मार्कर में यू विल गेट 4 पेजेस, ठीक है। अदरवाइज 15 मार्कर की क्वेश्चन रहते हैं हमारे पास, 20 मार्कर भी आपको देखने को मिलेंगे, वहां पर आपको 4 पेजेस देखने को मिलेंगे। दिस आई हैव ऑलरेडी डिस्कस्ड इन माय एनरिचमेंट क्लासेस, दैट्स व्हाई एनरिचमेंट क्लासेस आर, यू हैव टू डू इट आल्सो। If you are not watching it live, try to watch it in a recorded form. Over there I'll talk a lot of things, because beyond this particular content, when you will be absorbing all this content, you need that art of writing also in a time bound manner, right? So keep that in mind. So these are the things which we are going to settle down in this particular lecture. Let us start one by one, guys. ये आपने देख लिया, ये आप pause करके try to see this, and now right now you will not be much more focusing upon this. After this lecture you will be writing certain questions over here randomly, okay? It is up to you, and show me what you are writing, how you are writing, and what problems you faced, time bound manner in 2 pages, okay? Your note should be nice and well, as I have told you, that use the pausing mechanism. One topic is complete, then you pause it, you revise it, and then move ahead. This way you should do it, है ना? ठीक है guys, let us start. Over here, this is a cell, and this is a cell which was observed in 1665. ठीक है, 1665 it was observed as this, a rough structure like this you are seeing, and from this we are not able to know what is inside the cell, but this type of honeycomb structure we have observed, and not a particular scientist named as Robert Hooke, that you all must be knowing, written in every NCERT. And this was the microscope, primitive type of microscope at that point of time, which was invented. Okay, now you understand over here that there was certain evolution which I'm talking about, how it was happening in 1600, in 1800, and in 1900. There were scientists, important scientists, who were coming, and over here these gave certain basic tenets for this cell theory. Over here the research first made on bacteria, plants, and animals, you will be finding. Right. So over here, one scientist name who is the father of microbiology, that is Antony von Leeuwenhoek. He discovered, basically, he observed the type of cell like this. Today we know that this is a bacteria, this is a prokaryote. At that point of time, he visualized that, okay, he not visualized, but seen through his microscope, the primitive microscope at that point of time, which was invented by these people. They observed these particular thing, that this is something, this bacteria is something made up of like this. Whereas Robert Hooke, one important scientist, you will be finding, he will be observing the cork or the outer layer of the tree, that is a cork, and when he observed it under the microscopes, it also looked similar like this honeycomb like of structure which I have shown you just. When in animals, certain scientists over here, names are important, not important. When some scientists over here observed the animal cell, they are also finding like this. So that means they concluded something, that this particular, the basic, that over here cell is the basic structure of life. They concluded this. So this is a one theory part which is coming from here. In 1600 to 1700 time frame you will be seeing. Exact timeline is not needed, but in the PPT I have mentioned that. In 1800 you will be finding there were 2 important personalities who were coming to the forefront. In plants there was one person that is named as Schleiden, and the complete name you will be finding M.J. Schleiden. M.J. Schleiden, M.J. Schleiden was observing, he was a botanist, guys, and he was observing more about the plant cell and concluded more of the theories with respect to the plant cell. Whereas T. Sean was observing about or studying about the animal cell, as he was a botanist. At different time and space you will be finding these two were understanding about these 2 cells. But one thing they concluded by understanding, that plant cell and animal cell are different, but cells, all living beings, are consisted of cells. They have come up with this conclusion, that these cells are building up and giving the last shape as of a plant. These all cells are building up and giving the shape of an animal. So this they concluded over here. A lot of scientists will be there, but for the examination you will be remembering these 2, because these 2 are mentioned everywhere in your standard books. If you will start with BD Singh, Fundan Singh, or any particular book you will be picking up over here. समझ रहे हो? So as I want you to stress upon the lecture only, you don't need any other book, you don't need any other source. Class PPT, and you can explore more in the book. Same things will be written. Now you will be moving ahead, and in 1900 then there will be certain people who will be coming to the forefront, right? So over here there will be one personality, that is Robert Virtue, and in 1800, later 1800s, you will be finding, late 1800, you will be finding one personality who will be coming, that is Rudolph Virtue. And he cleared one confusion, guys. There was one confusion that from where cells are emerging. ये cells आ कहां से रहे हैं? What is the origin of these cells? So Rudolph Virtue gave one writing, and one of his important writing was the writing over here you will be finding in the form of, just wait guys, let me think about it. Hominis cellula a cellula. He concluded that these cells are emerging from a pre-existing cell. Cells are emerging from a pre-existing cell. That means their origin is not having, their origin is not happening out of nowhere. Their origin is happening because of a cell. Cell is dividing into two. In the bacteria, you will be finding binary fission. In the plants and animals, you will be finding mitosis and meiosis. Later on, we got to know about this. Later on, scientists gave this name. But ultimately, a rudimentary form of statement he gave. This is classical theory, cell theory, which you have understood over here. As per this classical cell theory, they give these basic tenets of cell, which still hold true when other scientists, when other modern scientists were experimenting with the cell, guys. This is the most fundamental aspect which you learn. That in 1600, 1700, they concluded some of the statements over here with respect to the cell. So what you will be remembering about. Now there are a lot of scientists who are engaged over here. Louis Pasteur also engaged over here. But again, we'll keep it short, keep it nice with respect to the examination. Till now on cell theory, question has not been asked, but they can ask question from here anytime. It would be your like out of nowhere, or once in a 4 or 5 years of time period, they might ask this particular aspect, and you will be the best candidate to solve it, because nobody focuses over here in much detail. Even if they will read, they will not be able to remember the Schleiden, Schwann, Rudolph Virtue, Antonin von Leeuwenhoek, Robert Hooke. But you remember it, and we'll be revising it again. Keep this in mind. This is the classical theory which you will be finding. Now let me show you. These are the people engaged over here. This is Robert Hooke, Leeuwenhoek. Over here, this is your Schleiden, Virtue, and your Schwann, right? So you will be finding these people. Classical theory, you will be, I have told you, Matthew Schleiden, Theodor Schwann. Schleiden focused upon that plants are made up of cells, animals are made up of cells. So they concluded that all living organisms are composed of one or more cells. ये आप याद रखोगे. When you will be writing classical theory, you contribution of scientists, you write like this. Then statements they gave, this you will be remembering. Second, the cell is the basic structural and functional unit of life. This was already been highlighted in 1600 by Robert Hooke and Antonin Leeuwenhoek. Limitation was they could not explain how cells was originating, and this particular dilemma was completed by Rudolph Virtue. Right. Also, no mention of cellular reproduction, lacked knowledge of organelles and cell chemistry. So later on, Virtue's addition, and he was Rudolph Virtue, added ominous cellula as cellula. That every cell arises from a pre-existing cell. This you have to remember by heart. And he added one more statement to this cell theory, that all cells arise only from pre-existing cell by cell division, not out of anywhere. They removed the idea of spontaneous generation. What is this idea of spontaneous? They removed the idea, because before that they were thinking that these cells are emerging from the combination of this abiotic and biotic. Biotic and abiotic means if there is a rock, ठीक है, ये rock है और यहाँ पर इसमें पानी गिर रहा है, यहाँ पर you will be finding rainfall is happening, air is there, and because of their mixture, they were saying that out of nowhere this cell is coming to existence. But this was not there. This was spontaneous. This is said to be spontaneous generation. So this is not happening. when they dig down deep, they understood that the cells are dividing over here. So cells are emerging from a pre-existing cell. Cells are not emerging from this abiogenesis or spontaneous generation. Understanding? So this particular statement is highlighting this. Next is introduce continuity of life also you will be finding. And modern cell theory, you will be finding that 3 statements are again put forward. And modern cell theory has been successful because of electron microscopy, molecular biology and biochemistry. And now we know that cells also contain hereditary information. ये 3 तो याद हो गई, अब ये 3 और याद करो. That they contain, अब ये आगे पढ़ोगे भी, you will be understanding this more ahead in the classes. Cell contains DNA that is passed during cell division. All cells basically the same in chemical composition and metabolic process. Energy flow occurs within the cell. Also over here you will be finding some more advancement. This also simpler in understanding, you can read. So classical cell theory and modern cell theory, I hope you are understanding. इनके बारे में और कुछ देखना है तो यहाँ पर written है already. Same information is written in the same way. Just read it out. You have to remember this only. Right? More modern cell, you will be finding more points over here. Modern cell theory. Right? So with respect to cell theory, I hope you have understood. Cell theory, you must have understood that the contribution of scientists over here, starting with Robert Hooke, Antonio Leeuwenhoek, then you will be finding M.J. Schleiden and Schwann. Then you will be finding the Rudolph Virchow addition. This thing you should remember. Classical cell theory, 3 points. Modern cell theory, add 3 more points. This is enough for any of the examination which you are going to see with respect to cell theory. Question has not been asked till now, but even if they will be asking you, this is, you will be in a better condition to solve this guys. ठीक है, अब आगे बढ़ते हैं. Lecture में you will be finding now the important things are coming, and when we got to know about the cell, as per the modern theory also, we have explored the cell really very well. And we explored the cell really very well. We understood that there are certain types of cell. And when we understood this, we got to know that there are majorly 2 types of cells which are present. That is one prokaryotic cell and one eukaryotic cell. And this I'll be telling you now in greater detail. So understand the clear difference between eukaryotes and prokaryotes, guys. Prokaryote cell, prokaryotes and eukaryotes. Prokaryotes are the type of cells which are very primitive. These are the first cells which came into existence. So they are primitive. Primitive. Why? Because over here they are in their very basic structure. You will be finding a closed like of this and a closed cell over here, and there will be no organelles which will be present. There will be no nucleus which will be present. Even if any organelle is present, that will not be membrane bound. So a very rudimentary type of structure you are seeing, and these type of things must have existed when the life was taking form. So the first creatures which would have come was the prokaryotic cells. And the major example of prokaryotes, you will be finding it in the form of bacteria, PPLOs. Right? These are organisms only you will be finding. So to classify all living beings on the planet Earth, one important classification criteria we take of are Whittaker's. So this classification is taken from Whittaker's classification of living beings. Right? So all living beings are classified by him into certain kingdoms. And how he has classified, let me tell you. He has classified that there are certain cells which are prokaryotic, which do not have any membrane, which do not have any organelles. So he termed this particular kingdom as Monera. So what a Monera kingdom will be having? All the prokaryotic organisms. Right? And the examples I'll be showing in the PPT also, bacteria, PPLO you can take. Then he saw other organisms or other cells which were basically unicellular. देखो यहाँ पर भी ये unicellular है. That means these organisms are having one particular cell only. Over here also one unique thing you observed, that there are unicellular organisms, but these cells are having a well-defined nucleus. ठीक है, न्यूक्लियस प्रेजेंट है. these particular cells are having your membrane bound organelles. अब ये membrane bound क्या है क्या नहीं है सब you will be understanding throughout this lecture. All the linkages will be happening. Membrane bound organelles are there. Means if there is a mitochondria, then there is membrane bound you will be finding. Membrane is there. ठीक है. So unicellular, अगर आपके पास दो unicellular, you have two type of unicellulars. One is not having nucleus, one is having nucleus. One is not having any organelles, one is having organelles over here. So you will be calling it as the kingdom of Protista. Both are having unicellular, but their features are different. So Protista, if I am saying, these are the type of unicellulars which are having well-defined nucleus, membrane-bound organelles. And the example over here which you will be writing will be blue-green algae, will be diatoms, will be protozoa. Whereas if you will be understanding that there are certain cells which he has found, which the scientists have found, and this person has classified it into that these are multicellular organisms. Now you plants are multicellular. So there is again one difference he noticed, that some organisms are having this cell wall present in their cell, and some organisms it not having cell wall. So those organisms which are not having cell wall are nothing but animals. So this from here one kingdom emerged, that is Animalia. So Animalia will have all one common feature, either reptiles, birds, fishes, you will be finding, or mammals. They will be having one common feature, that their cells will not be containing a cell wall. Now when we saw this cell wall cells which were containing these cell wall, further they divided it. And they divided it on the basis of that one cells who can carry out photosynthesis and those cells which cannot carry out photosynthesis. Photosynthesis means those who can make their own food. So those who can do photosynthesis, they clubbed them into this kingdom known as Plantae. And those who cannot do their photosynthesis, they clubbed them into fungi like mushroom and Plantae kingdom. You all know, which we are going to explore. So this area we are coming over here, guys. This particular chart you should remember. Again, I am telling you. Let me show you too. ये मैं आपको दिखाता हूँ ये chart. Just wait. देखो over here. Kingdoms: Monera, bacteria; Protista, algae, protozoa. Then over here Animalia, not containing cell wall. Containing cell wall but not containing photosynthesis, fungi, mushroom, yeast, mold. Containing photosynthesis, broadleaf tree, fern, mosses, and every plants which you are going to see. And then Plantae kingdom is also further divided, which we will be seeing throughout, like whenever there will be the right time to see that. Understanding? So by that way, you are understanding that whole organisms are either having prokaryotes and eukaryotes. And the eukaryotes origin is not independent. Somewhere from prokaryotes only they have become more complex. The organisms are going from a simpler unicellular to a multicellular organism. So, complexity कैसे बढ़ी? Throughout this evolution process, you will be finding it. Right? So, eukaryotes and prokaryotes, now see the difference. देखो, nucleus in prokaryotes is absent. What is nucleus? We have to study in detail. Next lecture will be all about this nucleus. So, there will be a, like, when you will be completing these starting 2-3 lectures, then there will be complete sync with respect to the topics, guys. Like, so those people who are non-bio, specifically I am saying for them that you try to watch it, all your doubts will be cleared. Okay, so nucleus, you will be finding that 2 hours lecture we have to take upon nucleus only. So over here nucleus is very important. This is the center part you will be finding. Nucleolus you will be finding over here. This is absent in prokaryotes, present in eukaryote, well defined, surrounded by nuclear membrane. Whereas the size of prokaryotes small. DNA over here, one important thing in prokaryotes, you will be finding that they are having a circular type of DNA like this. Whereas in the eukaryotic cells, in Plantae, animals, Animalia or fungi, you will be finding they are having a linear type of DNA. And this we will be exploring it also. That what is DNA, how this histone protein. Just remember that over here histone protein is also there. So what you have to do, that when we will be covering this whole cell biology and genetics, again you try to see this first lecture, because in first lecture you will be finding this histone proteins a bit. Those who are not from science background, ये आपको थोड़ा complicated लग सकता है. But over there, when I'll be teaching you about this histone proteins through animation also, then you will be understanding exactly what is a histone protein and what is its role. Very important role it plays. Then you will are finding membrane bound organelles absent over here in prokaryotes, present over in eukaryotes. Cell division, binary fission, mitosis, meiosis, again we have to understand it in cell cycle. So all these details, it is better that I will be telling you the differences again, right? So you will be understanding it again. Examples you try to remember: bacteria, cyanobacteria, plant, animal, fungi, protista over here. Okay, more differences you can see over here, but in a better way you will be understanding when you will be completing this lecture and furthermore 2-3 lectures. Okay, so this is another type of difference based upon the plant cell and animal cell. Most of the things you will be finding same, except vacuole, cell wall and chloroplast are exclusively found in a plant cell, and our subject is related to plants only, so we will be dwelling in plant cell more, not animal cell. Animal cell is having one thing, that is centriole, which is not present over there, and lysosome also you will be finding in animal cell only. Okay, so now we are dwelling upon the right, again the exact thing which is going to be asked in the examination, that is your cell organelles. And under cell organelles, the first thing which we are going to see is cell wall, very important. Then we are going to see cell membrane. Then we are going to see mitochondria, और सेल मेम्ब्रेन भी बोल सकते हैं इसको, प्लाज्मा मेम्ब्रेन भी बोला हुआ, प्लाज्मा मेम्ब्रेन, माइटोकॉन्ड्रिया. Then over here plastid, and under plastid we are going to see chloroplast. Then over here you will be finding ribosome. Then over here you will be finding endoplasmic reticulum and your Golgi apparatus. Very important. ठीक है, यहाँ पर ये चीजें समझनी है. We have to understand all this one by one. Let us speak and how we have to understand this. What should be the question over here? What is the structure, sir? What is the function over here? These two things should be highlighted in your notes clearly. So see over here, plant cell wall. Basically the most important over here will be starting with cell wall, and cell wall is a rigid outermost layer, of the plant cell, because cell wall is only found in plant cell over here, or in fungi also you will be finding. Now over here there are the structure which I am going to tell you. First of all, the structure you should know. In the exam also you will be drawing it as it is, guys. Okay, let me take some time to draw it. I am drawing it for you so that you can understand that how it should be drawn in the examination. You can draw it in a better way by taking more time, but again not required. Okay, like this. These are a different type of cells which I am forming over here, guys. Okay, this is 1 cell, 2 cell, 3 cell, 4 cell. Plant cell is this. Okay, and I have told you that outermost covering over here is a plant cell wall. So, but plant cell wall consists of basically 3 things over here. One is the intercellular membrane. Between the 2 cells there is a gap. So intercellular space is filled up by one thing that is called as a middle lamella. Middle lamella. This is one component you will be finding. Second component you will be finding with respect to cell wall is the primary cell wall. That this is the primary cell wall over here. This is the primary cell wall of this wall. And the second and the third component which you will be finding is the secondary cell wall, guys. Secondary cell wall is basically over here. This red one part. Secondary cell wall. So like this you will be finding over here secondary cell wall. Like this also you will be finding secondary cell wall. Secondary cell wall. Understanding? So this type of structure you are finding, that this is a basically cell wall, and cell wall structure is consisting of 3 things: middle lamella, primary cell wall, secondary cell wall. So first of all you will be understanding this. In the examination you can draw a diagram like this also. Like this also you can draw. No problem in this. Again, can you show me the secondary cell wall, primary cell wall? And the outer over here is must be intercellular space, and this must be filled up by middle lamella. Primary cell wall, secondary cell wall over here. Understanding? And then over here you will be finding that this cell wall, what this cell wall is doing, this is the cytoplasm. the plasm or the material inside the cell wall and outside the nucleus is known as cytoplasm. But if I include this nucleus also, this two things will be known as protoplasm. Understand the difference clearly. Cytoplasm is all the material which is present inside the cell wall and outside the nucleus. But if I'm saying protoplasm, that means it is including the stuff inside the nucleus also. So cytoplasm and protoplasm, two terminologies you should understand. This we will be seeing later on. Okay, but these are simpler things which you should understand. So I hope you are understanding this. Now you have to understand that this middle lamella, primary cell wall, secondary cell wall are made up of what? This you have to know in order to write one or two statements over here. So you will be understanding this middle lamella is made up of pectins you will be finding. Pectins are the compounds over here you will be finding over here, pectins. This primary cell wall is consisted mainly of cellulose, and this secondary cell wall is primarily consist of suberins, lignins, suberin, lignin and pectin also. ऐसा नहीं है कि pectin बाकी जगह नहीं होगा, होगा. Lignin, suberin, cutin also you will be finding over here. This you have to remember. So middle lamella is made up of pectin, primary cell wall cellulose, secondary cell wall suberin, lignin, cutin. And these substances are giving the property to the cell wall over here, because of the cellulose primary cell wall is soft. Because of suberin, lignin and cutin, the secondary cell wall is rigid, hard as compared to the primary cell wall. And this is giving the structure like this, guys. Understanding? Now let me show you how you will be writing and what is the function we will be discussing. This is just the structure right now. Let me show you the diagram also, how you will be showing you it in the examination. ये तो आपने ऐसे ही बनाना है. अभी डायग्राम में और दिखाता हूँ. अभी इसमें क्या आपको एक और चीज देखने को मिलेगा. You will be finding that over here there are certain structures like this also. Gaps like this you will be finding. And these gaps are for the intercellular exchange of materials, and these gaps are known as plasmodesmata. Nothing but plasmodesmata. Okay, now their role we will be understanding one by one. Let me show you the plant cell wall first of all. Can you see this? Observe this, guys. Alright guys, over here you must have seen the middle lamella over here. Then the primary cell wall over here. Primary cell wall, they are showing it in a wrong way. Over here primary cell wall is this outer one, then secondary cell wall this is inner one. Okay, so like this. Acha, they have shown it in the right way. Dekho, over here primary cell wall and secondary cell wall. Then plasma membrane, right now we are not focusing upon, we will be discussing it later on, don't worry. This is one diagram which you can make, but this is a 3D type of diagram. You don't have to make a 3D type of diagram. Let me show you another one. This type of diagram you can make, guys. I hope you can understand this. Plasmodesmata, these are the intercellular gaps you are seeing. Okay, plasmodesmata, secondary cell wall, primary cell wall, intercellular spaces, middle lamella. I hope you are understanding this. Again, see the broad structure for your understanding. I have added this. This is not required in the examination. See the primary cell wall over here. Then you will be going ahead secondary cell wall. And see, this is made up of lignin, hemicellulose. This is middle lamella is made up of pectin, right? Let me show you now. Because cell wall over here contains only in plants, absent in animal cell. These are non-living, that means dead, but produced and maintained by the living protoplasm. I hope you are understanding the protoplasm, including the nucleus also. They are having a rigid structure because it protects inner cell parts. Function we will be understanding. Their development happens from the phragmoplast, which involves all this cytoplasm and over here the protoplasm too. Now over here you see the structure. When you will be writing about the structure and they are asking you the structure of cell wall, you will be writing in the exact same manner in the examination. Middle lamella, it is the outermost layer of the plant cell wall, connects adjacent cell, composed of calcium, magnesium, pectate, and pectate is nothing but pectins. So either you write pectins or you write pectate, and kiske hain ye? Ca2 plus and Mg2 plus. So in your essential plant nutrients, I have told you that some of the uses of calcium and magnesium you will be understanding in paper 2. So now it is your job to highlight the importance of calcium and magnesium and write one line and highlight it, that this you can write in essential plant nutrients also. Understanding? Fourth is your all so-called intercellular substances or intercellular matrix, which I have told you. Then there is secondary structure. Then there is second important structure, that is primary cell wall, which is thin, elastic, and it lies between middle lamella and secondary cell wall, composed of cellulose. ये जो मैंने आपको बताया, ध्यान में रखो. Develops after middle lamella by deposition of cellulose, hemicellulose or pectin. Cellulose है, इसका मतलब ये नहीं है, और it doesn't mean that it will not be having hemicellulose or cellulose. What is cellulose? Cellulose, basically you are having certain biomolecules of life. ठीक है? Biomolecules. In biomolecules you are having carbohydrate, you are having fats, you are having proteins. In carbohydrates you will be finding that this is a monosaccharide chain, polysaccharide chain, and a polysaccharide, but you will be finding that this is nothing but cellulose. In fats you will be finding this is nothing but lipids, and proteins are formed by amino acid chain. ठीक है? तो biomolecule stuff you will be understanding in your science classes of GS, not over here. But as it is, you understand that this cellulose is made up of the carbohydrates, or you can say the polysaccharide chain. Okay? So this you must be understanding over here. Then secondary cell wall, this is the innermost layer, lies between a primary cell wall and plasma membrane, and it is compared to the primary cell wall thick. ध्यान रखो, primary cell wall thin है, अंदर वाला thick है, made up of cellulose microfibrils. This is important, microfibrils, and may contain lignin and suberin and cutin too. Understanding? Then there is one more part, that is plasmodesmata. Plasmodesmata are minute apertures in the cell wall which helps in intercellular transport. Connect cytoplasm of adjacent cell. दो सेल के cytoplasm connect कर रखा है इसने. More about plasmodesmata I will be telling you in the unit 4 plant physiology, when you will be understanding how the active transport and passive transport between the cell happens, how the need for food requirement, oxygen requirement, nutrient requirement, water requirement of a particular cell is being transmitted to other parts of the plant. So this you will be understanding more in separate class, but try to understand the basic structure कैसे है. वहां पर भी मैं आपको अगेन ये बताऊंगा, बट जल्दी मैं आपको ये वाली चीज बताऊंगा, राइट? Now functions, what is the functions of cell wall over here? These are written simply over here, what are the functions? But whenever you are writing the functions of cell wall or of any organelle, you try to think it from certain angle, that how it is giving the help in the structure part, how it is giving the help in the metabolism part, how it is helping it in the development part, how it is helping in the growth part. Like this you can remember it, right? One by one let us see how it is helping. Now most of the things you will be understanding more when you have read, or when you will be completing this whole cell biology and genetics. Then you will be learning about a lot of things. When you will be completing unit number 4 plant physio, then also you will be learning about the importance or functions of cell wall, because right now you are just starting, right? Those who have studied, they must be connecting, but those who are new, don't worry about the words which you are going to see now. देखो प्लांट सेल वॉल, हाउ इट हेल्प्स इन ग्रोथ. ग्रोथ में कैसे हेल्प करेगा? हाउ इट हेल्प्स दैट इट डिटरमाइंस सेल शेप एंड साइज. Over here the fixed shape and size which it is providing a mechanical strength over here. Also it gives a growth direction. This is important, that roots have to spread like this, not like this. Shoots have to spread like this. So this intimation is basically regulates growth direction. And how the plant growth is happening? Microfibril. How you will be understanding what is microfibril? I'll be telling you in the meiosis or mitosis part, right? जब cell cycle देखेंगे. So microfibril orientation guides directional growth. Example, root hairs or pollen tubes. How pollen tubes work? That when I'll be telling you about reproduction in plant breeding, you will be understanding that how pollen tubes make their way over here to the ovary. This you will be understanding later on. So अभी सिर्फ पढ़ो. यहाँ पर most of the things you will not be understanding as it is right now, but you will be understanding later in a much better way. As I have told you, all these units are connected. Again, some answers you will be getting later on. So it facilitates intercellular communication over here. Plasmodesmata in the wall allow transport of molecules, signals essential for coordinated growth. ठीक है? Next is in the plant development. One in growth plant development, you will be seeing that it helps in differentiation of tissues. How it get differentiated later on, like lignification means lignin compound, suberization means suberin compound, cutinization means cutin compound, which plant secondary cell wall is made up of. Pattern formation, cell to cell recognition happens over here. Just read the points right now, no need to go into detail of this right now at this point of time. Okay. Maintenance, it provides structural support, barrier functions, regulating water balance over here. How it happens later on you will be understanding. Again I'm saying reproduction, pollen development and germination, pollen tube growth, seed development, and fruit ripening and dispersal. This you will be understanding in plant breeding. Plant breeding, first chapter will be about the sexual and asexual reproduction in plants, and that I'll be telling you. Okay, so don't worry about it. So over here, plant cell wall, you are seeing that there is basically from 4 areas you have understood their functions. More than enough in plant cell wall. 2 things we have covered. Structure for your exam, 1 pager, explaining the primary cell wall, middle lamella and secondary cell wall, plasmodesmata. Then there is one more thing you have understood the functions with respect to growth, development, metabolism, and you will be finding mechanical support which it provides, and one more thing we have understood over here. This is the notes, final notes which you have made for plant cell wall. Again I am saying those people who are new and in their first attempt, they don't have to go for short writing right now. They will have to make extensive notes. Extensive का मतलब ये नहीं है that you have to copy the direct PPT into your notes. No, short, but you have to understand that what you have written. For those more than second attempt, third attempt, they are in there, you have to make, and they have also like gone for this particular foundation course. I hope this must be helping you. You have to go with the crux writing over here, because somewhere else you must have, because people also somewhere have studied then come over here, and now you must have studied over there too. So make the short notes over here. That means a 1 pager on a 1 page structure, on the next page function. That's it. Okay. Now let us see the second cell organelle, that is your cell membrane, guys. Over here, let us understand about the cell or plasma membrane. Again, what is the structure first of all, and then you will be understanding the function. Now with respect to structure, there are certain models which were provided by certain scientists over here. So these models are lipidoid model over here. You will be finding one sandwich model, and under sandwich model you will be finding there are two models came under this sandwich model, that is of unit model, and one is subunit model, and one last, that is fluid mosaic model, guys. Okay, so this for us, this plasma membrane is not simply this particular part inside a cell wall. After secondary cell wall, you will be seeing plasma membrane, and plasma membrane is not that simple, one layer. So that's why you will be finding these structures, these models have come time, and like you will be finding in the evolution of understanding plasma membrane from 1800 to 1900, you are going to see that these people came from 1900 onwards only, you will be seeing. And this fluid mosaic model 1972 you will be finding, right? So time and date I'll be telling you, but all this is happening in the 20th century, 1900 to 2000. All right. So over here, one by one, we have to study these models and what they said in 2 lines, and what were their limitation. Basically, you have to understand. Sandwich model was the most famous model, because of which fluid mosaic model came, and over here unit model and subunit model came. Try to understand. Okay, one by one, let us see. So this is your cell membrane, and to explain this particular part, let me explain you. This cell membrane you are seeing, this is cell wall you are understanding. Okay, now I am opening up this red part over here. In order to understand this red part, if you will be seeing this section, और ये वाला section देखो आप, अगर आप ये section देखोगे, you will be zooming it in, you will be finding the structure will be something like this. So what is this basically? This one molecule, if you will take out, you will be finding this is a lipid molecule, phospholipid. It is a phospholipid, right? And in this phospholipid, you will be finding that this part is a hydrophilic part, means which can attach with the water, hydrolover. And over here, you will be finding the hydrophobic part. That's why it is away from the water. So like this, they are attached. Right, and this is a chain over here you are seeing, and this is bilayer because one layer is this and second layer is this. So that's why you are going to find that this is a phospholipid layer, which this plasma membrane is made up of. Now you see this particular part, phospholipid, and connected with the hydrophobic tail. Over here, one more thing you will be finding that these are protein structure. Now, in order to understand the protein structure, which were there basically for selective membrane, that means this membrane is having the capacity to pass on certain chemicals inside and outside the cell. Again, listen, this particular layer is having the capability to let pass certain materials outside the cell to the inside and inside the cell to the outside. Selective permeability is there. How this selective permeability is coming? Because of the enzymes, because of the proteins which are present over here, which are acting as a channels. So the debate about this model is this only, that people were not able to tell that where these proteins are and how these proteins are embedded. The problem with these models was with respect to these proteins allocate. You will be finding that where the proteins are situated basically. I have told you that there is one lipid bilayer model, sandwich model, unit membrane, fluid mosaic, और यहाँ पर सब unit भी आता है. You have to remember the scientist name: Gorter and Grendel lipid, Danieli and Davison sandwich, Robertson unit membrane, Singer and Nicholson fluid mosaic, and subunit model over here you will be finding Green and Capaldi. So let me tell you now how you will be revising it, guys, or how you will be understanding it. First of all, revise तो बाद में करोगे. एक आपका मॉडल है लिपिड मॉडल. This lipid model explained over here by who was there? Green and what was the name? Sorry, Gorter over here. Gorter and Grandson. Right, this was the name, right? Grendel, Gorter and Grendel. They explained that this plasma membrane is made up of a phospholipid layer like this. They gave for the first time that this is a layer which is plasma membrane is made up of, and they are nowhere wrong in saying this. This is right. But they have not given in their explanation that where are these proteins situated in this particular model. They never talked about this model protein allocation over here. Never talked about. Right. So what you will be finding that that's why the second model came into place, which described that where these proteins are situated. And that's why this model is known as a sandwich model. How do you see the sandwich? There is a patties in between and two burgers, two buns above and below. So likewise, this sandwich model, which was basically suggested by Danieli and Davison. Danieli and Davison said that over here, there is no doubt that there is a phospholipid layer. Like this, there is a phospholipid layer. And they suggested that there is a protein layer also, but protein layer is embedded on from both the side on these phospholipid like this. Like this, there is a protein layer. Again, this was the limitation, because proteins you will be finding are not like this. Certain proteins are embedded inside the phospholipid layer also. So they have not given the explanation to the proteins which are integral part of the phospholipid layer also. They have given the information about the peripheral protein. Peripheral मतलब जो outside में हो, integral मतलब जो inside में हो. So they have not given information about the integral proteins over here. So next modification you will be finding in the form of unit model and subunit model. Unit model was given by Robertson and a subunit model was given by Green and Capaldi. Now these are nothing but the modification of sandwich only. So let me tell you one by one over here, guys. देखो lipid model. What is the feature? Propose that cell membrane is made up of two layers of phospholipid, hydrophilic and hydrophobic tail. Proteins are not included in the model. So that's why this is the limitation. that it could not explain selective permeability to ions and polar molecules, ignored proteins completely, provided no structural details beyond lipid arrangement. So that's why sandwich model came, which is also known as protein lipid protein layer. Bun, patty, bun. That's why sandwich. Who gave it? Danieli and Davison. Very important model. It was very prevalent in that particular time period. It suggested that lipid bilayer is coated on both sides by continuous protein layers like this, and between lipid layer. The proteins were thought to give stability and control permeability. The sandwich arrangement was believed to be universal. But what happened over here? Treated proteins as static layers, and they were later proven wrong. They were saying that these proteins are not static, but they are fluid. They are floating from one place to another place. And this was given by fluid mosaic model. So they could not accommodate hydrophobic membrane proteins embedded within the bilayer also. They did not talk about the integral proteins aspect. Or don't forget about this freezer fracture electron microscopy. These are the methods by which, microscopy methods by which they are being highlighted. These things are highlighted. Then J.D. Robertson came over here and gave this unit membrane model. He proposed that a trilaminar structure, 2 electron dense protein layers separated by a light lipid layer. Again explained already by the sandwich model. Then what extra thing they gave over here, that claimed that all biological membranes, plasma membrane, nuclear membrane, organelle membranes, which you are going to learn later on also, are having the same structure and thickness, which was proved wrong later on, because he overgeneralized and later found that membranes are having different composition and thickness. Did not address the fluidity, asymmetry and dynamic nature of the membrane, and still assumed that static proteins outside the bilayer rather than moving within it. That means they also did not talk about integral layer or integral proteins over here. Understanding? Then will come the subunit model provided by Green and Capaldi, also known as the subunit model. 1972 they gave. And what was as per this? They suggested that cell membrane is not a uniform continuous bilayer with proteins embedded in a fixed way. That means they are challenging the models proposed by UNIT, Robertson and sandwich model, Danieli and Davison. They suggested that it is a mosaic of discrete protein subunits arranged in a pattern with lipid molecules filling the gaps between these proteins. Means they highlighted that over here proteins are embedded like this, and in between that gaps these lipid layer is filling the gap like this. They gave this picture. Though this is an important finding, because upon this only one most accepted model you will be finding that fluid mosaic model was accepted for this plasma membrane. So in their view, lipids were not the continuous phase but were confined to the spaces between large and globular protein subunits. Don't try to think what is globular protein subunits. I'll be explaining you all. Just understand it in a simple way. Over here, first, who came? This lipid layer. They told us that only lipid layer this membrane is consisting of. When they are telling us this, then where are the proteins in this? So proteins were explained by sandwich model over here. Then subunit or this unit model is also emphasizing the same thing, but they were not saying, they are saying that these are static in nature, and they have not explained what about these proteins which are present inside the phospholipid over here. So subunit came and subunit said that over here this is not the structure. Basically proteins are discrete over here, and in between that the gaps are filled by phospholipid. So this was also challenged, and the ultimate model which came was fluid mosaic model, guys. Just wait. Fluid mosaic model by Singer and Nicholson. This you have to remember, 1972. Now they explained that membrane is fluid phospholipid bilayer. That means it is not static, it is fluid over here, and with the proteins embedded. Integral proteins के बारे में किसी ने बात नहीं करी. They are talking about integral or attached to the surface over here, peripheral. So lipids and many proteins can move laterally, giving the membrane fluidity. Explains asymmetry also. This you should be highlighted, that this explains asymmetry. Inner and outer leaflets differ in lipid and protein composition. This you will be understanding when I'll be showing you the image. and it allows dynamic processes like endocytosis, endocytosis means anything which is coming into the cell, anything which is going out of the cell is what is exocytosis, and cell signaling, what is the cell requirement over here. These are the instrument which were used, no need to remember it. Freeze fracture electron microscopy, FRAP over here. Again, there was some shortcomings over there in this model too, but this is the most accepted model for your this plasma membrane you will be finding. And let me show you how it looks like. Dekho, this is the way now you will be understanding. This particular एक structure को देखना अभी. See this only. This is your phospholipid layer over here. यहाँ पर देखो यहाँ पे. ठीक है. This is your one layer and continuous over here chain you are seeing of lipids and joined like this. And now in between this you will be finding which other models could not explain about the integral proteins. These are integral proteins written. This you will be highlighting in the exam. Then the proteins which are on the surface over here like this, like this, like this. These are known as peripheral protein. They have explained this also. Then rest of the cholesterol over here, these are also attached. This I have already explained, hydrophilic and hydrophobic end. ये आपको mention करना पड़ेगा. So in the exam you have to show this like over here, cell membrane in a neat way obviously. Then you show this integral protein. Explain this hydrophobic, hydrophilic end. Explain this particular protein over here and mark it as peripheral. This is our integral and put it into a box like this. Clean, neat and clean where you have shown all this diagram. Okay? And then explain the model. If they are asking you structure model, then you don't have to go ahead and explain these models in greater detail. You try to understand that how you will be writing it in short words. That this evolution of understanding of cell membrane started with the lipid theory given by Gotter and Grendel. This was modified and given more detail by the sandwich model given by Danieli and Davison, which was further improved by unit and subunit model of Green and Capaldi. And ultimately the most accepted model which we have with the limitation is a Fluid-Musyoc model. So from not understanding or giving importance to proteins and understanding that this is a fixed protein structure over here, our understanding from this Fluid-Musyoc model is now that these particular proteins are floating. Also, there are integral proteins which are embedded in the phospholipid layer. I hope you are understanding this. Okay, now you see the function, guys. Over here again you try to see जहाँ focus करा रहा हूँ वहाँ focus करना, otherwise it will be confusing. It is a phospholipid layer over here. ठीक है, these are the integral proteins. यहाँ पर देखो, these are the peripheral protein. कहीं लिखा होगा peripheral proteins यहाँ पर है. ये structure confusing है. Don't try to see this. ठीक है. अभी आपको मैंने बताना था साइटोप्लाज्म के फंक्शन ओवर है, प्रोटोप्लाज्म के फंक्शंस, राइट? Not protoplasm, but plasma membrane के function. Just wait. So functions over here you will be finding with respect to, just wait, है ना? Structural functions again. It will be helping you in giving the structure, physical boundary, maintain shell shape, anchoring sites, right? Anchoring sites means that over here it gives attachment points for the internally and extracellular matrix externally. This you will be understanding later on, because on the surface of plasma membrane only there will be certain channels, certain protein channels which will be there. Transport function which helps in selective permeability, passive transport, active transport. What are these? You will be understanding in unit number 4. Not right now. Passive means where energy is not required. Active means where energy is required. So over here pumps, ATP pumps are required and K positive. ये हम पढ़ेंगे सब उसमें. अभी सिर्फ एक बार रीड करो आप इसको. Communication functions, signal reception, signal transduction, cell to cell recognition. Again later on you will be understanding. ठीक है. Then there will be certain metabolic functions and protective and homeostatic functions will also be there. So just write it down in short about these functions. Details you will be understanding when you will be understanding this ATP synthesis process in respiration, mitochondria, chloroplast in the separate classes, when you will be over with photosynthesis, over with respiration, which are huge topics which we have to see, and very very interesting topics, guys. Okay. Now the next cell organelle. 2 organelles we have covered, guys, right? And 2 organelles are your cell wall and your but your plasma membrane, also known as cell membrane. Now we will be understanding the most important part over here, that is the mitochondria. Let me go to the blank page. Mitochondria over here. Very, very important, guys. Mitochondria, or let me just show you first the structure of mitochondria. This is the structure which you are going to see. ठीक है, mitochondria. Again, two things. What is the structure, sir, and what is the function? Mitochondria, there are certain scientist names you will be remembering, like Kolliker or Benda. I'll be telling you why they are important. Benda is the person who gave this terminology, that is mitochondria. है ना? And over here you will be finding that what mitochondria shapes like. This is the structure of mitochondria which I am drawing over here, guys. ठीक है? Mitochondria structure. Mitochondria, you will be finding that it is a double membrane structure. Double membrane structure. A very important part mitochondria plays because it is known as the powerhouse of the cell. And since childhood you must be knowing this. And why it is known as the powerhouse of the cells, and the answer you must have got that ATP gets synthesized over here. Now in this particular part, not over here, but in cell biology or in plant physiology, we will be understanding the process of respiration and how this ATP actually gets synthesized. We will be understanding it in the unit number 4. So right now just understand the structure. In detail function you will be understanding in unit number 4. Okay, so over here what you will be finding, this is the double membrane structure. So this is the outer membrane, and the inner membrane you will be finding somewhat like this. Like this. Like this, right? So this is the inner membrane, this is the outer membrane, this is the inner membrane, and this structure like this I have shown you, this are known as cristae. Cristae, this structure. Why? Because they have curved like this, because they can increase the surface area over here. And on these cristaes you will be finding one similar simple structure like this, small structure like this, and a lot of structures you will be finding. And these are nothing but your oxysomes. Oxysomes. In these oxysomes you will be finding the actually in ATP synthesis process happens. जहां पर ATP बनते हैं वो कहां बनते हैं? वो यहां पर बनते हैं. And the between this this space is known as mitochondrial space or perimitochondrial space. Perimitochondrial space. In some where you will be finding it written as intermembrane space. This is also right. This is cristae. This is inner membrane. This is outer membrane. And this is not enough now over here. You will be finding that this inner material of this mitochondria is known as matrix. And in this matrix, so this structure you will be highlighting, in this matrix you will be finding one circular DNA. That means mitochondria is also having its DNA. And this DNA is known as mitochondria. You must have heard that DNA is only contained in nucleus for the non-science students, but DNA is also contained in the mitochondria also, and in the chloroplast also you will be finding. Whereas in eukaryotes I have told you that there is a linear structure of DNA. In a prokaryotes there is a circular structure of DNA. So because of the presence of circular structure of DNA, this is giving us a hint that this mitochondria must have been there existing independently as a prokaryote. But later on, because of complex processes, it must have integrated into a multicellular organisms. That is one evidence which we are getting because of this circular DNA. This circular DNA, if there is a DNA, then there is one more organelle which will be present over here, whose job will be to produce protein. Because DNA is containing the code, and protein work where the code will be going to the ribosome. So over here you will be finding one ribosome is also present. In this ribosome we call it as 70S ribosome. And what is this 70S ribosome and other ribosomes? We will be clearing it out when we will be studying ribosome organelle. Just understand this. And there will be certain enzymes which will be present, which will be helping us in different processes which we are going to take place in mitochondria. So this is the structure which you must know. Matrix, inner membrane, cristae, oxysomes, outer membrane over here, it is a double membrane over here, right? Mitochondria now. See this, guys, over here. 1850s, the first notice intercellular granules by Kolliker. I have told you to remember Kolliker. And second, the name was given by C. Benda, mitochondria. It is present in almost all eukaryotic cells. and its number varies over here. Somewhere you will be finding it rod shape, somewhere oval shape, somewhere rod shape, filamentous shape. This is shape with respect to this. The structure, when you will be writing in the examination, you will be writing as it is. It is a double membrane system, outer membrane and inner membrane. An inner membrane is containing the folds inwards, that is cristae, which will help in ATP synthesis and electron transport chain. What is these? You will be understanding in the respiration chapter when we will be taking one full lecture on respiration. Understanding. Outer membrane contains smooth and permeable to small molecules, whereas inner membrane is selectively permeable. This also point you focus upon. Then here is a membrane composition. Membrane is formed of trilamellar, means tropoprotein layer sandwiching a lipid layer. Similar thing you will be finding. And then there will be intermembrane space, which I was talking you about. Intermembrane space. Let me show you another diagram of mitochondria. देखो ये intermembrane space है यहाँ पर. And this is, I was calling you it as perimitochondrial space भी बुलाते हैं हम इसको. And over here what will be happening? Oxidative phosphorylation. Just write it down. This is the whole process you will be understanding again in unit 4, respiration. Okay, then there is a mitochondrial matrix, and mitochondrial matrix you will be having matrix. What I was calling matrix is the inner material over here. Matrix, inner material, and in matrix you will be finding what? That site circular mitochondrial DNA, 70S ribosome. What is this? We'll be understanding later on, and certain enzymes which are needed for this different activities which are happening. No need to rectify it right now. Just write it down. Later on, when you will be doing other units, you will be understanding what is Krebs cycle, what is oxidation, what is phosphorylation. That you will be understanding later on. है ना? So this is the structure which you will be highlighting over here, guys. Now you see the functions of mitochondria over here, guys. The functions of mitochondria, again, just read it right now. No need to understand what is meant by this dehydrogenation, oxidative phosphorylation, decarboxylation. These all we have to understand in detail in the subsequent units. Okay, don't worry about this Krebs cycle, C3 cycle, C2 cycle, acetyl coenzyme. Everything we will be understand. So right now, just give it a look. Just read it down, pause the video, and at the right point of time. Those who have studied earlier this agriculture as their graduation subject, they must be understanding this. But those who are new, you don't have to worry about it. At the right point of time, we will be studying that, and again you will be coming over here and reading this particular slide or what you have written in your notes. Okay. So this is mitochondria over here. You will be finding powerhouse of the cell. One thing is written over here, endosymbiotic theory evidence. This is one important thing, that this particular mitochondria and your chloroplast give us one symbiotic theory. As per this symbiotic theory, it suggests that we have evolved from a prokaryote to a eukaryote. How do we know that? Because mitochondria is having its own DNA, ribosome, double membrane over here, and it is having the similarity to alpha protobacteria over here. Right? So this is one important point you will be finding, guys. Next, I'll be telling you about this is mitochondria only. Next, we will be moving forward and we will be learning about the plastids. Plastids are primarily present in the plant cell, whereas mitochondria present in both plant and animal cell. Plastids you will be finding primarily in a plant cell. And over here you will be finding there are two types of plastids which are containing. One is your chromoplasts, and another is your leucoplast. Chromoplast means the pigments in which pigments are present, and these pigments are giving the color basically. So certain chromoplasts will be giving a red color, certain chloroplasts will be giving the brown color, and certain chromoplast will be having this green pigment, that is chlorophyll pigment, which will be giving you the green color. This is our area of importance, and this particular chromoplast is having one subcategory known as chloroplast. And this is our area of importance because those who are having chloroplast are able to make food by the process of photosynthesis, by which this life is existing over here. Photosynthesis. So this you will be understanding over here. Let me take you to the plastids. So plastids I have told you over here. Now plastids you will be finding these are absent in animal, fungal and bacterial cells because they are not capable of generating their own food. The term was introduced by A.V. Schimper. Schimper you will be remembering. The type of plastids when you will be finding one chromoplast, colored plastids. Those pigments where chlorophyll A and B are present are known as chloro. Those pigments where फुकोजेंथिन, ये आपको याद करना है. फुकोजेंथिन pigment is present responsible for brown and yellow brown are basically pheoplast, and those pigments where red pigment are present, that is phycoerythrin. Fucoxanthin and phycoerythrin. देखो chlorophyll everybody remembers, but fucoxanthin and phycoerythrin very few will be remembering, and it will be giving you the rhodoplast, red color. Remember it by heart. Now there is one category I have told you, chromo. Under chromo you have chloro. Another category I was telling you, that is leuco. Leuco means colorless. There are no pigments which are present over here. So what is this leucoplast for? This leucoplast is for storage purposes. What you will be storing over here? Dekho, this leucoplast is for storage purpose now. What you will be storing over here? Either you will be storing the starch, fats or proteins. Because over here in chromoplast, the food is being prepared. Now food can be stored in the form of starch. Food can be form of fats, and food can be converted into the form of proteins. So all these things over here you will be finding under leucoplast, help in storage material. And how it will be storing? If it is storing starch, amyloplast. If it's storing lipids, elioplast. If it's storing protein, proteoplast. Amylo, elio and proteinoplast you will be remembering. Over here phycoerythrin and fucoxanthin is related to pheo and rhodo. This you have to remember over here. So plastids, if the structure you have to made over here, plastids are of two categories, that is chromo and leuco. Leuco is containing amylo, those who are storing fats. There is elio, that is who are containing this is starch, this is fat, and this is proteoplast, proteins. Chromo you are finding chloro, you are finding rhodo, and you are finding one more over here, that is pheo, right? So remember this. Now comes the most important thing to understand, the chloroplast. The chloroplast you have to understand, guys. This is the understanding you have to understand. What is chloroplast basically? And right now I am showing you the structure. And chloroplasts are the fundamental organelles over here which are responsible for photosynthesis, because of which this whole life is existing. The food which is being created is chloroplast. Again, chloroplast, when you will be understanding, it is a double membrane structure. थोड़ा सा और ढंग से बनाते हैं. Double membrane structure you are going to find over here. Double membrane, and its double membrane will be simply like this. It will not be having the curvatures like Christie, like of mitochondria. Right, and in the examination also I am drawing it for you guys only. Okay? And inside this, this is outer membrane, this is inner membrane, and in between this is a intercellular or intermembrane space. You can say perichloro space over here. Right, and this is the most important thing you will be finding over here, guys. And what are these? I'll be telling you. Very, very important things you are finding over here. This is the zoomed picture, right? A lot of things will be present over here. And what this unit is called as a whole, this unit is called as a whole as a grana. And out of this one disc-like structure which you are seeing over here, if I take it out, only one I am highlighting, the disc-like structure, it is made up of this disc-like structure. If I take it out, you will be finding that this is nothing but is known as thylakoid, nothing but terminologies. Thylakoid, it is a bun-shaped type of thing in which there will be certain material also inside this. And what is important material over here? There you will be finding the quantasomes in green color pigments over here. Chlorophyll, where it is present over here. So where the food will be making, the light will be striking over here and the reaction will be happening over here, guys. How this reaction will be happening, you will be learning it in photosynthesis in unit number 4. Understanding? So this is over here is known as quantasomes. Quantasomes, the group of several pigments you will be seeing over here. Also, again, these type of structures you will be finding, and these are connected by one channel known as Fret channel. So as per photosynthesis, you will be finding that photosynthesis is important because glucose is being made. And when glucose is being made over here, where it is being made. Over here you will be finding that glucose is being made. Later on, basically inside this chloroplast, this glucose will be made, guys. And to make this glucose in this particular space, this space you are seeing inside the chloroplast is known as stroma. So in stroma, in photosynthesis, I will be telling you there is one cycle which goes on, that is Calvin cycle. That also we will be understanding there. Right now, just understand the structure. Inside this stroma and inside the mitochondria, what you are finding? Matrix. Inside this stroma, you will be finding again a circular DNA. Circular DNA to prokaryotes mein milta hai. But here we are finding, that means it is also supporting the endosymbiotic theory, that we have evolved from a prokaryote to a eukaryote. When it is having its own DNA, it must be having its own ribosome also. And this ribosome is again a 70S ribosome, which is primarily found in eukaryotes. But we are eukaryotes, which are primarily found in prokaryotes. But we are eukaryotes. And in eukaryotes, in ribosome, I will be telling you, ribosome 80S is being found. And there you will be knowing the difference between 70S and 80S clearly. Don't worry about it. After this, we have to see ribosome only. And then there will be certain enzymes which will be responsible for this generation of glucose. You will be finding over here. This you don't have to mess it up in the exam. Keep it neat and clean over here. Just show that enzymes are present over here too. So this is the structure which you will be forming, guys. Now let me show you the diagram over here. Dekho, this is the structure. Jahan focus karwa raha hu, where I am letting you focus over there first of all. First you see the outer membrane, then you see the inner membrane, then you see the intermembrane over here. Outer, inner, intermembrane. Then we have opened it up and you will be finding this liquid is known as stroma. Then I have seen that there is one layer like this you will be finding, like this channel you will be finding. Dekho, this they have picked it up over here. So how these are containing? This is a thylakoid layer, and thylakoid layer is known as grana. So grana thylakoid is written over here. And then there will be thylakoid, one unit will be known as thylakoid. If you will open up thylakoid, there will be thylakoid space that is known as lumen and thylakoid membrane. Any doubt? Stroma, this you have seen, granum you have seen. Now you understand that in the stroma there will be circular DNA, nucleoid is written. There will be ribosome, ribosome is written. Understanding? Let me show you another diagram if I have. See this diagram, guys, clearly. How these channel are connected. Fred channel also. These two are connected with Fred channel. Okay. Granum, outer membrane, intermembrane, inner membrane, thylakoid space, chloroplast, DNA, stroma. Right? Anything else? If I have the other diagram also, let me show you. नहीं है यहाँ पर, तो आप देखो Fred channel यहीं कहीं लिखा होगा कहीं, है ना? These two must be connected by some channel over here. Not written, it's all right. But Fred channel also you take. So chloroplast distribution you should know. Shape I have told you, biconvex or plano-convex. Size and number. These are not that much important. Just read it out. Ultra structure is very important. Over here stroma I have. In stroma I have DNA, circular, 70RS, 70S ribosome, and RNA. In grana it is a stack of thylakoid. Light reaction happens over here, and this you will be understanding in greater detail in the photosynthesis. That means when I will be doing unit number 4, when we will be completing 3 units, you have to watch these lectures again. The most connection of this lecture with will be the other units. Okay? Rest of the lectures will not be having that much connections. I have told you that on this thylakoid quantasomes are present. Stroma lamella is nothing but Fred channel I was saying. So Fred channel is also known as stroma lamella, which is connecting to grana. Understanding? So this is one thing over here. Chloroplast, DNA already told you. Ribosome already told you. Okay, functions you will be understanding later on. What is light reaction, dark reaction, pigment storage. In unit number 4 you will be understanding more about it. Right. Now let us move ahead and let us understand ribosomes, guys. Ribosomes, again, structure and function. Ribosome, you all must be knowing that when you hear ribosome, it is important for what? For the protein synthesis, right? So ribosome over here you will be finding is consisting of 2 parts basically. One is rRNA and another one is proteins. That means if I want to form a ribosome, then I require 2 things, that is protein and rRNA. These 2 things will be going to the nucleus. And what is nucleus? All this we will be understanding. Nucleus. In nucleus, you will be having one nucleolus, and over there this ribosome will be getting synthesized. Abhi sirf samajh lo, just understand right now. Later on when you will be going deep into the nucleus also, then you will be understanding. Then again I'll be telling you over here ribosome is getting manufactured. When ribosome got manufactured, it is containing two units. One is the larger unit and one is the smaller unit. What we have found that in eukaryotes, in order to measure this, we have one unit to measure this ribosome basically, and there is different ribosomes which are present in the eukaryotes and prokaryotes. That means one more difference I am telling you between eukaryotes, prokaryotes, which earlier I didn't tell you. Why? Because the reason is coming right now. Over here what you will be finding that there is one Svedberg unit which is used over here. Svedberg unit. This is the unit for measuring the ribosome weight over here, sedimentation. It is a sedimentation unit. That's why in every ribosome you will be finding this S attached. So in eukaryotes you will be finding that 80S ribosome is found, whereas in prokaryotes this 70S ribosome is found. Now you must be understanding why in mitochondria and plastids, chloroplast, I was telling you 70S is present. But everywhere else this ribosome, either these are present on the endoplasmic reticulum surface or freely available over here. These are 80S in eukaryotes. This 80S is divided into larger subunit and smaller subunit. The larger subunit is known as over here, this will be 50S or sorry 60S and the smaller subunit will be 40S. Whereas if the structure is of prokaryotes, the ribosomes will be larger and smaller, and larger will be known as 50S and smaller will be known as 20S. Sorry, not 20S but 30S. Understanding? That means, देखो, simply you understand over here that this ribosome is nothing but a two structure over here. What do we say? This unit. And two structure is containing larger subunit and smaller subunit. Larger subunit in eukaryotes is basically total. पहले ध्यान में रखो. You understand that this is a 80S. Eukaryotes have 80S. Then you remember that larger one is 60S, smaller one is 40S. Similarly, larger and smaller in prokaryotes will be having this as 50S and this as 30S. अगर ये याद नहीं भी करना चलेगा, but this much eukaryotes you should remember. Understanding? Ribosome is built up of rRNA and proteins. This I'll be telling you later also. Understanding? And this is measured by S. This is Svedberg unit. Now the job of RNA is basically to create protein. How it create protein over here? Because in this particular, the protein synthesis, how protein is getting formed, this is the part of our next lecture or next few lecture. We are going to see this in animation way. I will be showing you. But over here what you will be seeing that this ribosome is consisting of one mRNA. What is this? In the next lecture you will be understanding. tRNA over here which is coming, and rRNA which is helping to form the ribosome. So ribosome structure is containing these three important particular molecules, or you can say the structure part of ribosome over here, right? So let me show you now ribosome in a better way. Where is ribosome? You see this ribosome, guys, large subunit and smaller subunit, and this is mRNA and this is a tRNA, right? Over here you must be understanding. If I am saying this is a eukaryote, that means this is of 60S and this is of 40S. If I am saying this is a prokaryote, that means this is a 50S and 30S. Combined it is 70. Combined this is 80S. Right? This you must be understanding, I hope. Now you understand ribosome. So ribosomes over here are present in all living cells containing, देखो, rRNA, that is ribosomal RNA. This RNA, what is type of RNA? What is difference between RNA, DNA? Next class will be understanding. Over here 60% you will be finding 40% protein. Absent in non-membranes over here. Absent membranes are absent over here in ribosome. Location endoplasmic reticulum, cytoplasm, mitochondria and chloroplast. I have already told you. ठीक है, डिस्कवरी 2000 में इनकी डिस्कवरी हुई थी. एंड टाइप्स ओवर हेयर based upon the sedimentation, that is basically Svedberg unit. And over here eukaryotes you will be finding 80S, prokaryotes you will be finding 70S. So 50s and 30s, 60s and 40s you will be finding over here. Right guys, now over here, these functional sites are also there on RNA, on this ribosome, like this A, P and E. Again, in the next class, I'll be telling you, and then you can add these sites also. Right now, no need to get cumbersome with this information. Okay, role is in mRNA, tRNA, rRNA. Again, I'll be telling you it in the next class over here. So just understand that this ribosome is playing a major role in the protein synthesis. And protein synthesis is our topic in the next class, when we will be understanding the difference between DNA and RNA. Right? So this way you will be understanding it. Next is your endoplasmic reticulum, guys. Endoplasmic reticulum. Very important. Endoplasmic reticulum. Endoplasmic reticulum, what you have to understand over here, that this is your cell. Inside the cell you are having a nucleus, and then you will be finding that there is a structure like this. A filamentous type of structure you will be finding in the examination. Also, you have to draw it like this, guys. The sole purpose of me drawing it is because of this. And somewhere you will be finding the structures as like this. Okay, like this. In a better way, you can draw it also. Okay, like this you will be finding. Now, this is the structure which is known as over here cisternae. This tube-like structure which you are shown, and these are interconnected over here, are known as cisternae. And sometimes on these particular cisternaes, these ribosomes are attached, and that's why the look is rough, and that's why this is known as the rough endoplasmic reticulum. Whereas on some there is no ribosome, so they will be known as smooth endoplasmic reticulum. And these are connected with the nucleus, so that whatever material will be coming out of the nucleus, for example, whatever code they must be sending, ribosome will be translating that code. And they will be packaging, or they will be forming the protein over here and sending it to next Golgi apparatus. So Golgi apparatus will be like this. Like this will be present in the cell. Golgi apparatus. So Golgi apparatus will be understanding next. So let me show you the structure of endoplasmic reticulum first, guys. देखो ये आपका endoplasmic reticulum. See, जहां पर फोकस करवा रहा हूं वहां फोकस करो. This is your nucleus. Now outside this nucleus, this connection you are seeing of this endoplasmic reticulum. On this you will be finding this ribosomes are attached, which are important for protein synthesis. ठीक है, just wait. Okay, so over here, this ribosome you will be finding. Now see, let me show you another structure first of all. This is the cisternae I have told you. Now new word is coming, cisternae. And this is the curve-like structure. Inside this you will be finding cisternal space. Right? Ribosome is attached on this, in this. And जहाँ पर ribosome attached नहीं है, smooth endoplasmic reticulum. I hope you are understanding. So the general features of endoplasmic reticulum is flattened sacs, branching tubules, but connected to each other. Alright, so over here you will be finding it is continuous with the double layered nuclear envelope. ये न्यूक्लियर एनवेलप की बात हो रही है कि ये डबल लेयर न्यूक्लियर एनवेलप के साथ कनेक्टेड है. ये खुद डबल लेयर्ड नहीं है. अंडरस्टैंड इट वेरी क्लियर केयरफुली, राइट? सो ओवर हियर दिस थिंग यू विल बी फाइंडिंग फंक्शन इज मैन्युफैक्चरिंग, प्रोसेस एंड ट्रांसपोर्ट्स बायोकेमिकल कंपाउंड्स. So on which endoplasmic reticulum ribosome are present, that means they will be doing the protein synthesis. And on which protein is not present, they will be doing fats, lipids, carbohydrates, starch synthesis. So over here you will be finding that on rough endoplasmic reticulum, production and processing of proteins for export is happening. Whereas on smooth endoplasmic reticulum, lipid synthesis, carbohydrate metabolism, detoxification of drugs and poisons happens. Right? Again you see this, lipid and carbohydrate, not protein. Protein is not over here. Also one more function like detoxification of drugs, steroid hormone synthesis, calcium ion storage. Over here again use of calcium. So these functions you are seeing of smooth endoplasmic reticulum. Whereas rough is engaged in the protein synthesis. I hope you are understanding this. What is glycosylation? I'll be telling you later on. In the right time, at the right time you will be seeing this. ठीक है, अंडरस्टैंडिंग गाइस. So over here you will be finding that your endoplasmic reticulum, first of all, this is a whole lot of things which is going on over here. This you don't have to understand, you are not a medical student, है ना. This you don't have to understand, but ultimately understand that over here this protein synthesis is happening. Not only protein synthesis but lipid synthesis is also happening, starch synthesis is also happening, hormone regulation is also happening over here, like the steroids will be formed over here, right? And then they will be taken out in the form of vesicles like this, and vesicular traffic, you see this particular part, and it will be going to the Golgi apparatus. Suppose over here Golgi apparatus is this, and see the difference between Golgi apparatus and mitochondria, or not mitochondria but endoplasmic reticulum, that they are not connected over here, but over here these are connected, right? So these will be going to the this particular part of Golgi apparatus, and Golgi apparatus is having two sides. One is which is near to endoplasmic reticulum is known as cis, and one is which is near to the plasma membrane away from the endoplasmic reticulum is known as trans. So based upon the position, this is cis, this is trans of Golgi apparatus. This will come with the Golgi apparatus, guys, not with over here, right? So I hope you are understanding. Just give it a go through right now, that it is a flattened disc, branching tubules, membrane is consisting on one large lumen, cisternal space जिसे हम बोलते हैं. अगर ये आप शो कर रहे हो तो इसके अंदर है क्या? Cisternal space. If thylakoid is there, what is under this? Thylakoid lumen, right? So lumen is a compound which is inside that. So overall, if you have to write the function on endoplasmic reticulum, either you can separate the function of smooth and rough, or you can write like this, that it gives the structural support, skeletal framework, intercellular transport, synthesis of proteins in RER, lipids, glycogen, steroids, hormones in SER, membrane source, nuclear envelope, all these things like cell membrane envelope will be basically supplied by this only. Detoxification happens over here, very important. In liver cells, you will be finding liver SER, a lot of neutralizing toxic substances. There will be process which will be happening. Impulse transmission and storage work it does, right? Next, you will be going to the Golgi body. Cis phase I have told you, which is near to endoplasmic reticulum. Trans phase I have told you, which is near to your, what do we say, this plasma membrane, है ना? ये plasma membrane है. अगर यहाँ पर साइड है तो ये आपका ट्रांस है, ये वाली साइड एंडोप्लाज्मिक रेक्टिकुलम के साइड है, राइट? Over here cisternae, these tubular like flattened disc like structure are known as cisternae, ठीक है? And over here vesicles are formed, and in these vesicles only you will be finding, देखो now what is happening over here, this is your nucleus, this is your endoplasmic reticulum connected like this, this is your Golgi apparatus like this. Whatever information, whatever proteins they will be marking over here right now, creating over here, will be going in the vesicle form to the Golgi apparatus, and Golgi apparatus has the work of like of a courier service, like they will be assessing it, then will be sending it, transporting it to the targeted site, that where it should go. Either this protein is for the cell wall maintenance, cell membrane maintenance, or it should go out of the cytoplasm. This all work of packaging and sorting is done by Golgi apparatus. ठीक है, so let me show you over here the function बाद में देखते हैं, पहले इसकी जनरल आप फीचर देख लो, that Golgi body, Golgi complex found in both the cells, 5 to 8 cup shaped membrane covered sacs which is known as cisternae. ये लिखना है stacked like deflated balloons, ठीक है? So over in animal cell 10 to 20 stacks you will be finding. It is discovered by Camillo Golgi, very important to learn, using his silver nitrate staining method. ठीक है? Over here you will be finding that its role is a distribution and a shipping department of the cell, where this particular protein has to go, where is this particular fat and starch has to go. This is decided by Golgi. Processes proteins and lipids from the endoplasmic reticulum and modifies it, sorts it, packages them for either secretion out of the cell, delivery to other organelles, or formation of lysosomes. In animal cell you will be finding lysosomes will be there, not in plant cell. For plant cell you are having a vacuole. Understanding this? So over here mechanism is transport vesicles from ER fuse with the cis phase of Golgi. Then in the Golgi cisternae modification happens. If there is any addition or removing of sugars, phosphate tagging they can do, so that wherever the targeted Golgi body or the targeted protein should go, it can go. Synthesis of some macro molecules also happen. So one is modification, second is synthesis also happens of some macro molecules like polysaccharides, pectins, and it exits through the trans phase, enters through the cis phase, exits through the trans phase over here to the final destinations. I hope you are understanding this. So structural polarity, cis phase, trans phase, you will be understanding. Near ER, near plasma, this you will be understanding easily. Specific function, protein modification, lipid modification, polysaccharide synthesis. So you try to remember that it is not only about packaging and sorting Golgi bodies. One synthesis also happens, and this synthesis is of polysaccharide, which is used in the pectin and hemicellulose, which are further used in the cell wall of plants, right? And sorting and tagging also happen over here. So this all you have studied the cell organelles now, guys. This is all we have to study in this. So we started with the cell theory. I hope you are remembering the classical cell theory and the modern cell theory. Classical, I have told you to remember like 1600, 1800 and 1900 over here. In classicals, you will be finding some scientists named as Von Hoek, Leon Hoek, Robert Hooke, M.J. Schleiden, T. Schwann and Rudolf Virchow. Then after understanding cell theory, you must have understood the cell type, the eukaryotes versus prokaryotes, plant cell versus animal cell. More you will be understanding when these particular units will be getting over, this particular cell biology will be getting over. Then you must have understood about the cell organelles over here now. And cell organelles over here, we started with the cell wall, cell membrane, mitochondria, plasma membrane तो हो चुका, cell membrane को plasma membrane बोलोगे, chloroplast, ribosome, endoplasmic reticulum and Golgi apparatus, guys. All right, this is your cell organelle structure and function. What you have learned over here, structure and function. Okay, now what you will be doing, you have to attend the enrichment session of this, where I will be doing active recalling with you all. ठीक है, जो नहीं कर पाएगा, जो later on देखेगा, वो recording जरूर देखें. और active recall, when I'm asking something, you try to recall it at that point of time too. So now let me just tell you that how, after doing this lecture, try to attempt the question like plant cell wall, chloroplast, endoplasmic reticulum, Golgi apparatus. This you try to do. Okay, so cell structure and function we have tried to cover over here holistically. Now in the next lecture, or wherever the time will be right, next lecture we will be focusing upon the nucleus, because nucleus, one important cell organelle is left. DNA, RNA we will be understanding about, chromosome we will be understanding about, and in that lecture only I'll be telling you about the cell cycle too, after understanding nucleus, guys. Okay guys, so thank you so much. This was all for this particular lecture. I hope you will be revising it and then proceeding ahead with the next lecture. Thank you so much.